Here's the latest audio research news from the transmitter. Mitochondrial landscape shifts across human brain by Claudia Lopez Lareda. The number of mitochondria and their function vary across brain regions and cell types, according to the MitoBrain map, the first detailed atlas of the cell's energy factories. The map, derived from a biochemical and molecular analysis of mitochondria in adult human brain tissue, reveals a heterogeneous bioenergetic landscape that was hidden in previous neuroimaging studies, says Martin Pickard, associate professor of behavioral medicine at Columbia University, who led the new work. Until now, neuroscientists tapped PET and MRI to try to understand mitochondria biology throughout the brain. But these imaging approaches lack granular resolution, and the features they capture, such as blood flow, are typically only proxies for energy production. We don't know much yet about the energetics of the brain from imaging, says Valentin Riedel, a researcher at Friedrich Alexander University and at Technical University Munich, who was not involved in the new map. Gray matter has more than 50% more mitochondria than white matter, and different brain regions show varying levels of respiratory capacity, according to the new findings, published last month in Nature. The mitochondrial variability and local specialization also suggest regionally distinct energetic strategies, Riedel says. In particular, brain regions that arose later in evolution, such as cortical areas, have mitochondria that display a particularly high capacity for ATP production, which cells use for fuel. The results jibe with neuroimaging evidence of a metabolic gradient in the brain, published in 2023. The more recent human brain regions had a higher energy turnover than the older, evolutionarily established brain areas, says Riedel, who led the 2023 work. Before, we could only do that on the macro scale, he says. The new study controlled for regional variations in the number and density of neurons, giving a more accurate picture of the energy-producing capacity of the mitochondria. The results are perfectly matching, he says. To create the map, Pickard and his team needed to partition brain tissue into small cubes, or voxels. After a few failed trials, which included trying to slice through room temperature tissue using diamond wire, they realized the tissue needed to be frozen first. So the team bought a laser engraver and set it up in a walk-in freezer. They managed to cut up a brain slice containing both cortical and subcortical areas into 703 3mm cubes. The team then characterized each voxel's mitochondrial density, mitochondrial enzyme activity, respiratory capacity, and gene expression levels via single-cell RNA sequencing. They neatly covered everything from structure to function, Riedel says. Even though the white matter had many fewer mitochondria than the gray matter, Pickard says he was still surprised by the amount it had, because white matter is not that energetically demanding. Within the gray matter, phylogenetically older brain regions, such as the brainstem, had lower respiratory capacity than newer ones, such as cortical areas. This picture aligns with the idea that newer brain regions may have evolved to sustain the increased energy costs needed for the development of higher-order cognitive functions in humans, Pickard says. Even across cell types, not all mitochondria look the same. For example, endothelial cells in the brain showed the highest expression of energy-transforming enzymes compared with other cell types. This result suggests that mitochondria in the endothelial cells might be more active, but that is more a puzzle than an answer because it could also indicate they generate high levels of reactive oxygen species, says Fahmid Haider, professor of radiology and biomedical imaging and of biomedical engineering at Yale University, who was not involved in the study. Some mitochondria might even be specialized for specific functions, such as burning fat. One of the dogmas in the field is that the brain burns only glucose, not lipids, Pickard points out. That's true for neurons, but it's definitely not true for other cell types, he says. There's so many neurons that if you look at it as a whole, the brain doesn't seem like it's burning lipids, but there are some cell types that are actually capable of doing that. To try to bridge the gap between the molecular and imaging worlds, Pickard's team created a computational model to predict mitochondrial profiles from neuroimaging data. They first aligned 539 voxels from the postmortem brain slice, the subset, with complete mitochondrial data onto averaged brain scan data from an MRI dataset. They then used 80% of the voxels to train the model to match the mitochondrial features to the MRI metrics and found that it accurately predicted those features in the remaining 20% of voxels. In another test, The model accurately predicted mitochondrial features the team had measured in a second brain slice from the same brain donor. Other researchers can use the freely available atlas to ask interesting questions, Riedel says. We have the option to look in our brain imaging space now into which areas are more or less efficient or more or less active. And you can start comparing that to patterns that are particularly important. The study is a great technological feat, but it is based on post-mortem tissue, which uses energy differently depending on how it is processed, Hyder says. Once you take the mitochondria out of the brain, their structure begins to deform. And the findings need to be validated with direct measures of metabolism because the features the team measured, such as mitochondrial enzymes, are indirect and can only predict energy production, he adds. Further validation and studies can then also reveal which imaging parameters are related to which aspects of mitochondrial signaling activity, Riedel says. But this is just the first version of the map, based on 1 3 mm thick section of an individual human brain, limiting the generalizability of the findings, Pickard says. The team is working on its next iteration, which aims to average measures from 500 brains from both female and male donors across different ages. Mapping the mitochondrial infrastructure is the first step towards understanding the brain as an energetic system and understanding how that manifests into important problems in neuroscience, Pickard says. This audio was created using artificial intelligence. Stay up to date with the latest neuroscience news and perspectives at thetransmitter.org. Follow Audio Research News on Spotify, YouTube, or wherever you get your podcasts.